doesn't matter at this moment. So module 2, right, of the 2024 paper, figure 4 show, well, with this rendering of figure 4, it tries to show uh, a photomicrograph of the transverse section of the xylem vessels of a plant, right? So we have that there, the xylem. And we must draw 4 adjoining xylem vessels as shown in figure 4, right? Now, can I trust you guys to be able to draw this? Tell me yes or no if you can draw it. If no, you know that you need to go back to refresh how to draw them. Alright? So you need to refresh. And it's a biological drawing, so I would find my magnification because they give me 100 micrometers right here. So I would assume, uh-huh, all drawings must be of biological nature. Oh yeah, if you don't have that, you can't really do the micro magnification like that per se. But yeah, so you calculate your magnification, everything for your drawing, and you put your title and you draw your nice sexy xylem vessels, right? And you make sure that you don't shade or anything like that, and you keep right closed lines, right, etc. You try to be as true to this picture as possible, right? Make sure your pencil is sharpened and ready to draw. You don't sketch at all. No, no, no. Impossible. These drawing rules are something that are really traumatizing to me because when I did TSET biology, I did my first ever lab and I was so excited in fourth form. And my teacher threw the lab book through the window and I was on top of the fourth form block. So that's how you know that you actually have to learn through trauma sometimes. But yeah, so you have to make sure that you have a perfect biological drawing here for that. And remember your magnification because your picture does provide it. So remember to look for that, right? Use your ruler, find your scale, and use that. All right. So the next question: xylem vessels are adapted to transport water to the rest of the plant. Oh my gosh, what a revelation! After the water enters through the root, look immaculate. Complete table two by stating how each of the named structural features of xylem vessels allows them rather to carry out water transport in a plant. Alright? Now we're seeing here that structural feature here, right? It's lack cell contents. So how does this feature helps the tissue to carry out its function? How? I thought if you lack cell material then that would be a problem. Go ahead. The master of water without any metabolic interference. That sounds really good. Alright, so that would be a response that I would put there. Any other responses? Repeat? No, I don't think surface area would be a good thing to put here. Mhm. So seeing that it's no metabolic interference, right? You're gonna end up with a hollow lumen, right? And the hollow lumen allows for ease of flow, unobstructed flow. Unobstructed flow of water, right? Of the water there. So we need to note these different things. So unobstructed flow, right? We're gonna have faster conduction as it relates to the mass flow, right? Through cohesion and tension. We're gonna look at stuff like that, right? Moves more effectively through these empty areas. Alright? Continuous tube, what does help? What does that help with? It's continuous. The sea pores break down, right? So no more seed plates are left and we'll end up with continuous tubes. How does that assist? Okay, we could do the same thing on obstructed flow. Anything else? Okay. Right, so continuous columns there. So that's fine. Right? So when we have that entire adhesion, cohesion happening, the water wants to move in that continuous chain. So we don't have to force water through sieve plates or break it up through sieve tubes, anything like that. Alright? Lovely. The narrow lumen between 0.01 to 2 millimeters. So facilitates what? Capillarity. Okay, so facilitates capillarity. Lovely. Anything else? No? I mean, you don't have to write. It's not a lot you're going to write. Right? So it can facilitate capillarity, and you can speak to that. All right. Lignin being in the walls. Let me check the chat to make sure I'm not missing anybody. My apologies. Go ahead, Sunil. Shanil, raise your hand. Okay, lovely. To the hydrostatic pressure. Anything else? Mm-hmm. So we have that adaptation to negative pressure, right? So we have that the empty lignin reinforced xylem, right, would not only withstand stress, right, but assist in adhesion, right? So we could look at that. You could look at the cohesion adhesion theory or a cohesion tension theory rather, right? So looking at that there. Oh, let me add this person to the meeting. All right, lovely. So pits inside walls. What do pits allow for? Go ahead. Lovely. Full stop. Lateral movement of water. Right? So looking at that. So no cytoplasm, nothing like that required. Right? Just have lateral movement of water there. Allowing the efficient hydration of parenchymal and sclerenchymal tissues. But I don't think you're required to state that. Right? So we have that there. So in the with and then well, I mean I'll just put it there, a piece of it, right? So it leads to effective hydration or efficient hydration because all areas of the plant need to be hydrated. So we have that lateral movement of water, so it covers everything on that level of the plant. Alright? Efficient hydration rather. Hydration. It also somewhat prevent it does prevent embolisms as well. Right? Why I want to give more responses or more functions for the more difficult ones because a lot of persons tend to forget these uses. It also prevents embolisms, right? Which means air bubbles. So it prevents embolisms via the pit membrane. Well, not the pit membrane. Would it be the pit membrane? The pit membrane traps air bubbles, right? And the tormented pits are what we call the conifers. Hopefully, you guys know the entire physiology of the plants like that, right? But the conifers would have valve-like structures that seal off air bubbles, right? So when I went through biology, it was spoke to the striations and the conifers and the pit membranes and these type of things. So I have more information for the more difficult features because everybody knows what a continuous tube is used for, right? But we need to have more information for the more difficult parts, right? Cell wall thickenings with form rings or spirals. What are those thickenings called? Lignification. We're speaking about different forms of lignification. Any responses? Let me check the chat. Okay, provide structural support. That's what we're saying. So it increases the rigidity, right? So it increases rigidity. We're also speaking about what else? What else? What else? What else? So it increases rigidity. Speaking directly to spirals and rings, the rings and spirals provide reinforcement, or rather reinforcement, right? So looking at that as it relates to the rigidity of the structure. Anything else? It prevents collapse under tension. What else can we state about this? It aids in flexibility and growth as well. How does it aid in flexibility of growth and growth? How? Do we know how? So the thickened regions of the xylem would prevent growth, right? The unthickened regions, right, would actually promote growth there. So the unthickened regions are where we're gonna have cell wall stretch and the growth of new organs in the plant from those areas. But I don't think that's what it's necessarily speaking about. So I guess we do have to include that. But everything here should be fine. Any questions or concerns with the content of this page at all? If we're all comfortable, you can drop a numeral 1 in the chat so we can move on. Or if you'd like to add something or query something, please raise your hand and go. So we're all fine? If you have a question, you can go in. No? Go ahead. Hmm. The fast flow of water upwards. So if it's narrower, yes, that would be the case. That would be the case. Alright? So you could speak about that. It, along with osmotic pressure, would facilitate the fast movement of water. Alright, so that really now depends on the turgor pressure or the osmotic pressure. Alright? But yes. Lovely. Water is transported in a continuous column from the roots to the leaves of the plant. Describe how cohesion and capillarity aid in the ascent of water from stem to leaf. Anybody would like to start with cohesion? Yes. Hmm. Okay, so that is the case. All right. So it sounds like you're having a bit of trouble formulating the answer, but you understand the content that is required. Lovely. So we have water molecules, right? So we're saying that the water molecules, right? I'm gonna just use basic language, I guess. Stick together. I can't use um any other word there. Let's stick together. Yes. Mm-hmm. Yes. Beautiful. I'm to be very honest. I am very biased as it relates to any answer that incorporates more chemistry. Um, but yes. So look at that. But that is the case, right? So looking at the sticking together of the water via hydrogen bonding, that's what cohesion in the water speaks to, right? So no, it's for that. That is exactly what forms the continuous column. Continuous column. All right. So I have that there in the Xylem tubes. So you're gonna have something like that. So water transported is to describe how cohesion aids in the ascent of water. We're seeing water molecules like that, right? It in combination with adhesion would that would help as well. That adhesion would stick to the. Oh my gosh, I forgot to add something. Where is the lignified walls? As a as a thought about it, I have to come back to the question to add it here. Alright? But what's happening here, right? Well also the fact that the xylem was what was the xylem? Are hydrophobic, not true? Aren't they hydrophobic? Not hydrophobic, hydrophilic rather. My bad. Lovely. Alright, so I have that there. So speaking about the adhesion there, right? But I didn't want to necessarily you can state a little bit of that here, but the question really asked about cohesion, right? So you could speak about that, right? And that is what creates the um that this cohesion is what allows for water molecules to successfully move through the plant or gain meaningful ascent throughout the plant through transpiration and pull. Alright? What is capillarity now? And what does capillarity have to do with this? Okay. Right, so through the narrow tubes, and these are what we call the xylem tracheids. You guys heard that term before? Trichids or tracheids, depending on how the person pronounces it. Tracheids. That is how it's spelled. I don't know why this is flagging it. Right, so it allows water to rise against gravity. Right? So it allows water to rise against gravity due to what? Due to what? Adhesion. Due to surface tension. What else you can say? Looking at those things, right? And we could also speak to math, right? I don't know why I'm doing this to you guys. But we can also speak to the fact the thinner the tube, the higher the water rises, right? Not true. So we can note that height is inverse proportional to the radius of the tube. So the smaller the narrower the xylem, the greater the height the water can travel, right? So height is inverse proportional to the radius of the tube. This is truly, really and truly what capillarity is. Right? Capillarity is this expression that height is inverse proportional to radius. Right? So that allows for the water to move against gravity through up the plant. Alright? Hopefully me adding in these concepts don't confuse anything for anybody. Alright, but that is that there. B. Sucrose is transported from regions in the plant called sources to regions called sinks. Wow, what a revelation. The companion cells in the phloem are adapted for the loading of sucrose from the source. Explain how one structural feature of the companion cells enables them to carry out their role in sucrose transport. Sucrose transport. Alright? So, any brave individual would like to separate themselves from the rest of the pack and answer this question. Mm-hmm. Mm-hmm. Okay, lovely. Thank you for that response. Really comprehensive. Alright? Anyone else? There are more than one, and we will state more than one here. We're going to try to like raise hands because I hear that persons are speaking over others, right? Which is not intended. Alright, so you guys just raise your hands. Allow everybody a chance to deliberate. Alright? Okay, go ahead. Mm-hmm. Okay, so it allows numerous pathways looking at the simplistic path pathway. So it allows simplicity or simplistic movement. So looking at not only simplistic pathway, but it also allows what a direct pathway, not true? Okay, direct transfer. I'm gonna use a different word. I don't know. I want to change up the usage like it's an English exam. Alright, so I have that there. So we're looking at the simplistic pathway, right? So between the companion cells and the sieve tubes, right? And we're also the facilitation of the direct transfer of sucrose, right? And other types of molecules there. Right? We have anything else? I don't. Well, we also have a nucleus. I guess for metabolic support. So we could look at the nucleus, right? So should I? I don't think I should look at this, because we're gonna look at the production of ribosomes, and those ribosomes would actually create the proteins that are needed for all sort of transfer. I don't think we need to go so far. Um, so I don't need to go so far. But one thing we could note. Go ahead. Lovely. So we have these transmembrane proteins that facilitate you said the apoplastic movement, right? Hmm. Right, so it supports the apoplastic movement of sucrose there. So apoplastic, right? So we have the apoplastic movement being provided through the being provided through the transmembrane proteins, and we also can note that the component cells have a folded membrane, right? So it provides more surface area packed with these transmembrane proteins that allow apoplastic rather movement. Alright? And now we have the plasmodesmata that gives us our symplastic pathway. Right? And then we also have the organelles like the mitochondria that allows the active transport of these things, right? So we must note these things. Everything should be fine here. 3 marks. Alright? A carrot is a taproot. How interesting. That can be used in vegetative propagation. Sure, even though the question has nothing to do with vegetative propagation, you bring it up anyways. Explain how a carrot can act as both a source and a sink at different times. So we're going to start with source. Hmm. So what's a source? Could you define for us what's a source before we begin? Sure. Hmm. Okay. So when photosynthetic conditions are not ideal, right? We're saying that the carrot can be used as a source. Do we all agree with this so far? Can be used as a source. Because the leaves are not actively producing sucrose, then we must get the sucrose from the storage unit of the plant, which is the carrot, the root here, right? The root of the carrot. So we have that there. So what are we gonna be doing here now? So when photosynthetic conditions are not ideal, or we could state when during stress of the plant, when the plant is stressing, so during stress, right? Or a reproductive phase. We speak about many other things. Stress, right? So we could look at so what would stress be like? Now that we're thinking of it, so when the plant, so stress would actually link into some of the photosynthetic conditions as well. Plants can undergo stress through there, right? Or we could look at the plants being attacked by different organisms and stuff like that through predation and stuff like that, right? So these different things can cause the plant to stress. Alright. So within any other, I just naming 3 different times in which the card can act as a source. Alright. But now I need someone to explain here now. So when, okay, go ahead. Somebody raised their hand. My bad. I think someone raised their hand. No? Alrighty. Alrighty. So we have that there, right? So looking at that as a source, right? And we're saying now that when that is happening in the plant now, right? Sucrose is loaded from the carrot to the phloem for metabolic processes elsewhere. Right? Any other? Well, there's just one more left. Phloem. I spelled it wrong. All right. So now, how does it act as a sink now? So looking at now, what's the time during growth periods? All right. So it acts as a sink. So we're looking at sucrose being loaded into the carrot. Lovely, right? So we're gonna have that there, right? This is for use in metabolic processes, really and truly. So if we have growth or anything like that, we're gonna have the use of sucrose in the root for the growth, elongation, development of the root, right? We could also state in times where we have when nutrients are plentiful, right? So the carrot is a storage organ, right? Right? So its nutrients are also plentiful, right? So just looking at that. So this is not obviously the presentation of how you'd write the response, right? But it's just giving the points that you'd use to answer this. Alright? Any questions or concerns? Let me check the chat. Make sure I'm not missing anything per se. Okay. Alrighty, so this is a 3 mark question, so most can answer that. Alrighty. So moving on, C. Plants and mammals rather both transport fluids. Table 3 shows a comparison of the transport systems of plants and mammals. Table 3, right? So I have that table there. So the feature that we're looking at now. So transport system in plants we have xylem and phloem, and mammals have hearts and blood vessels, right? Features for the fluid now that plants move are xylem sap and phloem sap. Mammals have lymph and blood, right? Then we have the substances transported for plants is water, minerals, and organic nutrients. Mammals know it's oxygen, carbon dioxide, glucose, and water. That is kind of reductive though because we do have other organic nutrients, minerals, and stuff like that moving in the blood as well. But it's fine. Alright, we're gonna take Cape's word for it. Explain the differences between plant and mammalian transport systems as it relates to the following. You must present your response in complete paragraphs. So what we're doing, we're explaining the differences. Alright, so the first one is the nutrients, not nutrient requirement, the energy requirement rather. Right, so that's the first one. Then mechanism of movement of fluids, then control of the rate of flow of fluids. Let me just check how many is 4, or is that it? That's the last one? Yes. So the control of the rate of flow of fluids. Go ahead. All right, so we're looking at the mammals, right? So it's saying the heart and blood vessels, and we're saying that it will require you have an ATP requirement, right? So they depend on active energy consuming pumps, right? So looking at the mammalians depend on ATP, right? So in the mammalians now, right, that entire system, right, mammalians generally depend. So what we're saying now, holistic, is that they depend on energy consuming compounds, right? So looking at something like that. So you just need to explain that, right? So okay, so while know what the plants have then, what's the difference now? What's the plants now? Hmm. Are you sure about what you said about the xylem? The xylem requires energy? I want to know, you know. Did somebody raise their hand? But I want to know, I want to know, I want to know. The xylem is dead, not true. Okay, so what we're talking about now, right? The phloem needs well does active loading and unloading, right? Using pressure flow. So we require so in plants we use pressure flow, right? For the phloem, which is different from mammals, right? And then for the xylem now, right, we're gonna be having passive physical forces for the xylem. So the passive physical forces, these are your osmotic pressure, your turgor pressure, your capillarity, your cohesion, your adhesion. Right? They don't need to consume ATP and these different things. Right? While your arteries and your veins would need to consume these things. Right? So looking at that. So they don't require that. Alright? As it relates to now the mechanism for the movement of fluids now. What are the key differences there now? All right. So plants, right? So plants are literally what I've stated there now, right? So since it's non-living, xylem are non-living, non-living, right? What we're gonna have here, it's the fluid flow is driven mainly. I have to put mainly because bio is necessarily absolute, right? But mainly by evaporation, not even evaporation, transpiration. And physics. That is what the plant uses. It uses transpiration and greater physics to move. Right? That's what it uses. Not true. Yes or no? That is the case. So up here, we can speak about the energy requirement here, right? Because the xylem is dead, right? The plants do use ATP for loading and those type of things as it relates to the phloems, right? But we're just looking at general transportation of fluids, right? In that case, right, the major organs that transport fluids, right, would be the xylem, and the xylem are non-living. So the energy requirement there, you have to speak about the gross difference in the energy requirement for fluid flow in mammals versus plants, right? For the mechanism of movement now, we're looking at the plant and primarily using physics. We're using pressure, we're using cohesion and adhesion, right? We're using transpiration pull, which is our osmotic pressure, right? While in animals or mammals specifically, what do mammals use? They use muscular pumps, right? As in the heart. Go ahead. Oh, okay. Okay, so we could say that. Okay, lovely. So for the mammals, we're looking at this is really a throwback. So heterotrophic nutrition, and then for the plants it would be autotrophic, right? Yeah, but the thing is, no, we could note those things, but it won't primarily strengthen the argument, right? Because the question wasn't necessarily speaking to where they got the energy from, right? But the energy requirements in their circulatory systems, right? So you could, I guess, you could mention it, right? But it won't necessarily add to the question per se, all right? That's just my observation there, all right? As relates to mechanic, wait, did this entire text box delete after I was writing in there? No, it's still there. Okay, so I have that there. For mammals, no. Wait, it may be here. Somewhere here. It's not here. Okay, so for mammals, we're gonna have hearts, so muscular pumps. What else could we state for mammals? The key differences within that. We're gonna in mammals they use pressure gradients, which is actually a similarity because of the pressure, the mass flow hypothesis thing, the pressure flow hypothesis rather. So we could also state in plants, right? So in the xylem they use that, but for the phloem we use the pressure flow hypothesis, right? So in mammals, can you add what we use? We also have vasodilation and constriction. That helps with movement in veins actually versus the arteries, which we should know. All right. So just need to note the differences there. The rate of control. Well, I mean the control of the rate of flow of the fluids. How do we have that now? So we control the rate of flow for the plant, right? What we use environmental. I need to state specifically it's not just environmental, but we use external environmental factors. Those things will affect the rate of flow of fluid to the plant, right? And they also use osmotic. I wouldn't say osmotic signals per se, but osmotic pressure. Let's say that, all right? So those things determine the rate of flow of fluids in the plant, the transport system. So looking at stomatal regulation, we're looking at Environmental factors such as light, humidity, wind, right? That is what we're looking at. No, right? I'm not hearing anybody. So are we on track? Okay, lovely. But for mammals now, what do mammals use? Mammals will utilize the autonomic nervous system. So use the nervous system. We use also we do use hormonal control, right? So you could think about those things. So the rate of fluid flow in mammals are controlled using um neuro well nerves, right? The neurological system there, as well as the hormonal control, right? Go ahead. Basically, basically that is the case. All right. So lovely. So that brings us to the end of module 2 of the 2024